Hello everybody, my name is Stephen Wilmes. I'm a senior scientist at Miltenyi Biotec, and today I would like to welcome you to the CGTI webinar on flow cytometric assays for CAR T-cell manufacturing and patient immunomonitoring. But before we really dive into the different topics, I would like to quickly introduce Miltenyi Biotec as a company. So Miltenyi was founded in 1989 in Germany. We have now more than 3,500 employees worldwide from at least 50 different nationalities. A quarter of them is working in R&D. We are well recognized in scientific publications, having more than 17,000 products already on the market, and many of these products are being used in cell therapy procedures. But we are also using our own cell products on patients. Right now, more than 6,000 patients per year. And finally, we are serving our customers worldwide, either with direct sales or including distributors. The headquarters of Miltenyi Biotec is in Bergisch Gladbach in Germany, but we have also facilities worldwide. Here, I would just like to highlight our main sites for cell processing and manufacturing in Bergisch Gladbach and in San Jose, and sites for development and processing of lentiviral vectors in Tetero and in Gaithersburg. So the agenda for today's webinar is as follows. I'll start with a quick introduction, followed by flow assays for manufacturing and immunomonitoring. Then we continue with different strategies for improving and facilitating your own flow assays. Then we will hear about CAR Detection Reagents from Miltenyi, followed by advanced applications where you can use CAR Detection Reagents, for example, in imaging and cell separation applications. And in the end, we will have a question and answer session. So I guess you're probably all well familiar with the basic principle of CAR T-cell therapy. Nevertheless, I will give you a very quick heads up. So CARs, so chimeric antigen receptors, they are designed to recognize tumor-associated antigens and bind this way the tumor cells with the scFVs. So this could be here, for example, the very famous CD19 antigen. Then signal transduction and co-stimulation triggers the T-cell activation process. And in the end, the activated T-cells, they attack and destroy tumor cells by various means. CAR T-cells derive from patient materials, and they need to be manufactured in a highly controlled process. So this starts with the patient material itself, then followed by the cell separation, cell activation, the transduction, expansion, the formulation and filling, and finally with the cell product. And essentially for all the different stages of manufacturing, flow assays are the ideal choice. So starting at the immunophenotyping, then the different in-process controls throughout the manufacturing, in the end the quality control, but last but not least also the patient immunomonitoring after back infusion of the cell product into the patient. And for these different flow assays, we like to use the MaxQuant analyzer. MaxQuant analyzers, they are state-of-the-art standardized platforms for flow cytometry. They come in different flavors, offering either different parameters, lasers or plate settings, but all of them, they have a couple of key features in common. For example, the automated calibration and compensation. They come with auto-labeling, offer chill racks and integrated plate processing. They also offer automated housekeeping for essential buffers and liquids. They even come with a pre-enrichment unit, which you can use to enrich sparsely abundant cell populations before you run your flow cytometric experiment. They also offer absolute cell counting, and in the end, the software, also including the express modes and output tables, they are compliant for communications to regulatory bodies such as the FDA. So now we're coming to the different strategies for reducing variations in flow cytometry reagents. Of course, the success of a flow cytometry experiment very much depends on the quality of the used reagents, especially the flow antibodies. So Miltenyi is taking advantage of the rare affinity technology. The rare antibodies, they are essentially recombinant antibodies that all derive from a defined DNA sequence. They are produced under standardized conditions, ensuring a very high lot-to-lot consistency. Also, they only require a single isotype control for your experimental setup. And since the Fc region was engineered and mutated, also no longer Fc receptor blocking is required for your experiment. Another sophisticated strategy for reducing the variations in your flow experiment would be the use of what we call stain express. So the idea is to use dried antibody cocktails. They all come pre-formulated. They're all based on the reaffinity technology of what we just heard before. And in the end, it means we have less handling steps and thereby less variations during your assay setup. So how can this experimentally look like? So you start with your barcoded stain express tube. You can just simply add your sample and mix it. Incubate for 10 minutes at room temperature. If required, you can add a lysis step for your red blood cells. And in the end, you just proceed with your flow cytometer. Also at the level of analysis and reporting, there are certain pitfalls to care about. When dealing with biological samples, we always have to take into account sample variability. So here's just a quick example of 2 batches of CAR T cells gated via CD3 positive cells. And here the task is to identify the CAR positive cells, so the transduction efficiency, by the help of the CAR detection reagent. So if we just focus for the moment on donor 1 and we set the gate for the transduction efficiency here for these CAR positive cells, this all looks nice. But if we try to adjust or transfer this gating strategy now for the second donor, it's very obvious that this strategy fails, simply because the starting material was a different one. So if, as a consequence now, you have to manually adjust these gates, this is of course a source of sample variation, which you ideally try to avoid. And a possible way to solve this problem is the use of a gating adjustment tool, which is based on algorithms. This way you can avoid variations caused by the human factor and thereby standardize also your data analysis. Our MaxQuant analyzers are equipped with a MaxQuantify software, and this software provides different types of express modes, which all allow fast and reproducible sample analysis. And of course, there's more than just a single express mode, so we have tailored express mode packages and also CAR T cell panels. And in the end, this is time saving, and this is the little icon that we chose for the express modes. So an average MaxQuant user takes about 17 minutes per panel, and the express modes, they just do this in 30 seconds just with a click of one button. It's also very reproducible because it's algorithm based. So here's a little experiment where absolute numbers of T cells were counted for 3 biological samples, either done with algorithm based analysis by the express modes or by manual gating. And I think you can acknowledge that when the gating was done manually, there's a much wider distribution when directly compared to the express mode results. Now I would like to highlight one example of express mode gating for you. So here's a CD19 CAR T cell sample during manufacturing on which we ran the transduction efficiency express mode. So after standard scatter settings, we just focus on CD45 positive cells. We exclude dead cells by the use of 7-AAD, live dead marker, and just continue with the viable cells. Then from the viable cells, we only take a look on the CD3 positive cells. And in the next gate, we can already determine the transduction efficiency among these CD3 positive cells with the help of the CD19 CAR Detection Reagent. And furthermore, you can also get the distribution of the CD4 or CD8 cells among either the CAR positive cells or the CAR negative cells. And in the end, what you also get is a full output table for all the different gates here. And probably the most important information for you would be the fraction of CD19 CAR positive cells either in the viable cell fraction or in the viable CD3 positive fraction. And this will be relevant in the end for your formulation and the dose of your drug product. There are even more established CAR T-cell panels offered by Miltenyi. Just to name a few of them, for example, the CAR T persistence for differentiation, activation, proliferation, exhaustion, fitness, or even effector cytokine production. And all these panels have been extensively tested by our experts, and they all come, of course, with a list of materials, with a proper staining protocol, and with a gating strategy. And if you want to look it up by yourself, you can just follow this web page here. And all these strategies that I was just mentioning have one purpose. They can make your life in the lab a lot easier. If you just have a quick look on the standard CAR T-cell manufacturing process, starting at day 0 here with sample prep, and on day 12, the cell harvest and the formulation of your drug product, you see that at least on 5 different occasions, you will need to read out the immune cell composition, here highlighted by panel A. And on top of that, you will also have to regularly check on the CAR T-cell transduction efficiency by panel B. And if you now can take advantage of our rare affinity technology, and even using stain express cocktails combined with fully automated analysis by the express modes, this will not only speed up your experiments substantially, it will also boost the reproducibility of your experiments. But the journey of the CAR T-cells continues after back infusion into the patient, because now we're interested in reading out the key parameters for immunomonitoring, and again this will be done by flow cytometry. And sample material could be either whole blood or bone marrow from the patient. So we would like to learn about the expansion of the CAR T-cells, not only in the absolute counts, but also about the ratio between CD4 and CD8 positive cells. We also would like to know about the efficacy of the treatment. So for example, whether or not we observe tumor clearance, or also whether we see target antigen loss. And also would like to phenotypically characterize the CAR T cells. Again, not only absolute counts, but also the differentiation state, whether we see central memory cells, effector memory cells. We want to learn about the activation state, whether or not they are exhausted, and we also would like to functionally characterize them, for example, by reading out the intracellular cytokines, such as interferon gamma. And for many of the flow panels that you just saw, you need to monitor the fractions of CAR-positive cells. To do this, you need to have reliable CAR detection reagents. And CAR detection reagents, so the name is kind of self-explanatory, but they are tools to probe CAR T cells in your samples, mostly by flow cytometry, but we will also hear about imaging later on. But how does this work in detail? So when it comes to basic academic research, you can rely on the co-expression of indirect markers, such as LNGFR. So this receptor can be either equipped with a GFP, or it can harbor a tag that you can use to target by other flow antibodies. But the clear disadvantage is you can't really apply these strategies when it comes to clinical trials. Likewise, also the use of generic binders is not really favored in clinical trials because of the same reason, and the lack of specificity. Also protein L, the staining performance is usually not really great for this type of reagent. So at Miltenyi Biotec, we favor 2 different types of strategies for CAR detection reagents. The first one is based on the antigen itself. So we recombinantly express the antigens, fused to an Fc part, and biotinylate them. And in a second step, you can target these CAR detection reagents by anti-biotin antibodies. And these come in different colors. Here, for example, VioBright 515, but you also can get different other fluorescence readout colors. Alternatively, you can also choose anti-idiotype antibodies. These are rare antibodies that were raised to specifically bind the scFv of the CAR. Both strategies for CAR detection come with their own advantages. For For the antigen-based CAR detection reagents, they are able to target all CARs that are specific for a chosen antigen, for example CD19. So if you have to switch your system to a different CAR, the antigen-based CAR detection reagents would still be able to bind and target also this new CAR. So here, the CAR detection through multiple binding sites is possible. It also shows that you have functional CAR folding, because essentially what you're doing is you're mimicking the interaction of your CAR T cell with binding the antigens of a tumor cell. Also, you get a high flexibility in your panel design, because you can, just as an example, you can exchange the emission color of the secondary stain reagent. And right now we are offering antigens for CD19, BCMA, CD22 and CD33, but there are also more to come in our pipeline. For the anti-idiotypes, here we get the CAR detection just via a unique binding site. We have here the advantage that we can use a one-step staining, also possible with different emission colors. And this is also compatible with applications beyond flow cytometry. And right now, we're offering two different anti-idiotypes that both target CD19 FMC63 CARs, the REA1297 and the REA1298, and both of them are targeting different epitopes on the same CAR. All our CAR detection reagents on the market take advantage of this engineered Fc region that you have already heard about when introducing the reaffinity antibodies. So there's very little unspecific background signal in your flow assays due to this unique engineered and mutated Fc region. On top of that, all these CAR detection reagents, they exhibit an excellent lot-to-lot consistency and also purity. They come already in a liquid form, so they are ready to use formulated for your streamlined workflows. And maybe most importantly, they have been really extensively tested, also on real CAR T cells and not only on beads. Here is a little example for highlighting the high lot-to-lot consistency of our CD19 CAR detection reagent. So these are flow experiments performed on CD19 CAR T cells. 4 lots of CD19 CAR detection reagent have been tested with each 3 technical replicates. And you may appreciate a very nice signal to background, which barely differs among all the tested samples. So as a consequence, all tested lots measure exactly the same frequency of CAR-positive cells. A hallmark of our CAR detection reagents is the very high specificity and sensitivity. Particularly during immunomonitoring experiments, it is absolutely critical that you can reliably detect even sparse fractions of CAR-positive cells. And this is something that can be tested in advance on lyased whole blood samples of healthy donors, because these samples, they should not show any signs for CAR-positive cells. And here in this example, we have tested the antigen-based BCMA CAR detection reagent. And while you can see in this sample without any staining, also pretty much no signal for CAR-positive cells. Also, when treated with the BCMA CAR detection reagent from Miltenyi, this channel is basically empty. However, when directly compared to 2 BCMA CAR detection reagents from different vendors, you can pretty much see a clear CAR-positive fraction in these channels, even though there shouldn't be any. And this makes these CAR detection reagents pretty unattractive for immunomonitoring purposes. We have tested the staining performance of all our antigen-based CAR detection reagents on primary CAR T cells. So a good separation can be seen here for the CD19 CAR detection reagent, for BCMA, for CD22. But some of our detection reagents have already been successfully tested on CAR NK cells as well, for example the CD3. Again, for all our antigens, we do see very little to no unspecific background on lyased whole blood samples. And that means if you have a bright signal, so a high mean fluorescence intensity, accompanied with a low background signal, this results in a high staining index. That's what we are aiming for. And this is very consistent also from lot to lot, here shown for an example of 2 CD19 CAR detection reagent lots that have been tested on 7 donors of lysed whole blood. Here, the unspecific background is even below 0.1% of CD3 positive cells. The same standards in terms of staining performance are set for our anti-idiotype antibodies. So here I would like to quickly introduce this REA-1297. So this anti-idiotype antibody targets CD19 CARs that are based on this FMC63 scFv. This antibody comes in different fluorescence colors, such as PE, APC, Virobite 515, but also as a biotin conjugate. And I hope you can appreciate that for all of these conjugates, you can easily distinguish CAR-negative and CAR-positive cells. And again, the unspecific background is for all these conjugates as low as for our antigens, showing that both of our CAR detection reagent strategies are suitable for immunomonitoring purposes. Now I would like to switch gears and demonstrate what else can be done with CAR detection reagents beyond flow assays. We and others have successfully tested that the REA-1297 anti-idiotype antibody can be used in imaging applications. And this spatial information is relevant not only if you want to check for tumor infiltration of your CAR T cells, you can even functionally phenotype them. And we did this using our Maxima imaging system. That's the instrument down here. And the Maxima is a phenotyping platform that allows staining cycles of more than 100 cellular markers in your fixed tissue samples. And it always works like this. You start with your staining, then you image, and then you erase the signal, then you continue with the next marker. And here we use this technique on mice harboring human tumors that were treated with different human CAR T cells. So in these fresh frozen slices, they were PFA fixed, DAPI stained, and also stained for different markers, also including CD45 to identify human immune cells, but also REA-1297 to find your CAR T cells. And here in this image, you can easily see the outline of your CAR T cells. And here in the merge, CD45 in blue and CD19 in red, you can appreciate that only a fraction of the CD45 positive cells is actually also positive for your CD19 CAR. And that's pretty much also what you would expect. In contrast, control CAR T cells that were not based on the FMC63, so the FMC63 is a target for the REA-1297, these were not stained at all, again highlighting the specificity of this REA antibody. So one nice feature of the Maxima is also the software, so the MaxIQ view. So using spatial segmentation, you can in the end display your images pretty much like a flow assay and do lots of functional analysis with this. And here, just as an example, you can do a gating-based detection of CAR-positive cells. So here on this axis you have the CD45 signal, and here is the signal for the REA-1297, and you can set the gate here. And in the end, in this window, only these cells light up that you have been selected in this gate. CAR detection reagents can also be used to enrich sparsely abundant CAR T cell samples. This can be incredibly helpful if your analysis requires a much higher purity than your original sample offers. So you can label your CAR T cells with biotin-related CAR detection reagents. Here's a CD19 antigen, for example, and then you can mix these cells with anti-biotin microbeads here. And if you then apply magnetic activated cell sorting, so classic MAX technology, only your CAR T cells will remain bound on the column. And the moment you remove the magnetic field here, you can elute your enriched sample. And this is a little example for enrichment of CD19 CAR T cells from a whole blood sample with as little as 0.2% of CAR positive cells as a starting point. The flow through is essentially empty for CAR T cells, and in the eluted fraction there's a very nice enrichment visible. Using an LS column, you get in this example about 80% of purity and a recovery of still more than 40%. But if required, you can also further boost the purity by subsequent max steps. Now I want to conclude this webinar and summarize all the different ideas for optimizing your flow assays. So you've heard about the max quant analyzer, our flow cytometer, the rare affinity technology for optimizing our flow reagents, these stain express tubes, so the conveniently dried antibody cocktails, and also about the fully automatized flow analysis by express mode gating. And in the end, I also demonstrated the different applications for our CAR detection reagents, for flow cytometry, for imaging, and also for cell separation. And with this, I would like to thank you for your attention, and I'm happy to take your questions now in the following question and answer session.